afternoon, everyone. Thank you for coming. It is indeed a privilege that the last of our special lecture series prior to opening the medical school is our own Dr. Kevin Tracey. You know that we've had lectures on each of our values, and one of our values is innovation and being dedicated to not ever being stuck in the status quo. That means however we used to do it, we're not going to do it. Even if we think it's good, we know it can always be better. And there's no area where it's more important to have a mindset of innovation than as you approach the link between diseases for which treatments are not yet satisfactory and linking the basic science to tackling those diseases and ultimately coming up with clinical strategies and clinical tools that change the course of human disease. And that is the mission of the Feinstein Institute. So Kevin Tracey, who went to Boston College and Boston University, and then decided to move down to the New York region, is a neurosurgeon. And there are not a lot of neurosurgeons who head basic science research institutes, or even basic translational science research institutes. But Kevin became infatuated with one of the unsolved mysteries of medicine today, which is sepsis, and the maladaptation of the immune response when it becomes destructive instead of protective. And from that work, I think you're going to be led down a story of inductive and deductive reasoning and fascinating serendipity in terms of observations that no one had ever made and explanations that reset the thinking of the immune system of sepsis and how we should approach it. And I think that Kevin is not only the leader of our research enterprise, he didn't even know it, but he is the Senior Associate Dean for Research at this medical school.
I knew that.
He knew it. He knew it. We did put his name on a door that is only a closet because we want him to think he has an office here. But we just claim we don't have the key to the door, so he doesn't know that there's only brooms behind him. Kevin, welcome, and thank you for coming.
Good afternoon, good evening. Thank you, Larry, very much for inviting me. I will tell you that I've never given a talk on innovation ever, anywhere, and I had to look it up and prepare for it. And so what I'd like to do is spend a few minutes talking to you about what I've learned about innovation for the first few slides, and then spend the remaining time talking about what we've tried to do innovatively in science. And I will leave you with the take-home point that innovation occurs in science as a result of a violent collision with hypothesis. Hypothesis in science, I think we're all trained, is something that you think of, this must be the answer. It's a declarative statement, and then you do experiments to explore whether or not it's true. But to me, that's fine, and it's important that science gets done that way. But the real excitement comes when, in thinking about the hypothesis, you actually begin to imagine the way the world is put together. And what if it was actually put together differently than the book said? What if it was actually assembled and worked differently than the world tends to believe? And that's where the fun part of science comes. And I think I'll show you some examples of that have occurred historically and in recent times. And it becomes very, very important. I mean, to be honest, I'll confess, I'm very interested in doing science because I want to make drugs. I want to make drugs and devices that will make people feel better. I went into medicine because it would give me better opportunities to do science to make people feel better. And so what has come out of this are a series of shots on goal Larry and I were just talking about, and hopefully some of these will be translated into the therapies of the future. And you can decide for yourselves whether this is a result of hypothesis testing, accident, or innovation, or all three. So to begin, I saw this lecture was open to the public. This is not a straight scientific talk, and I decided I better check and see what is innovation? What does the world define innovation as today? And in fact, a Google search reveals that it's about money. The business world has hijacked the word completely. It's about products and development. And this bothered me because I like money. I don't love money. I really like money, but I don't love money. In fact, looking around this room, I would say none of you love it either. You'd be the other school across campus. You really love money. We like money. But why is this all about money? And I think it probably has its relatively recent origins. I'm not an entomologist or an English professor, but I suspect that the change in the word in our times was heavily influenced by Peter Drucker, this leader in management and strategy and business consulting, who really has been credited with being instrumental in how business functions in the world today. And he said business has only two basic functions, marketing and innovation. And that was sometime 30 years ago. And really the focus has come on innovation related to products, making money, changing your business. And now look, to be honest, if we're going to change my medicine's practice, there's going to be products. And that's going to require lots of money. But I think the approach that's interesting in science today is to recognize that as a tool, not the driving force. So I like money, I don't love money. I love doing science. I love inventing. I love discovery. And I love doing that kind of innovation. So let's take a step back and redefine or think about what innovation was more in the history of science. And we can begin really with the NIH's definition. Now here I was pleasantly surprised because I was speaking to a colleague at Harvard a couple of days ago, and I said, there's a problem with innovation, it's all about money and products. He goes, oh yeah, I guess it is. He goes, I get an email, he goes, I'm at Harvard, he goes, I get 10 emails today telling me to be innovative, and I don't really know what it is. He goes, maybe the NIH defined it. I said, well, they should have defined it. I said, because it's one of the criteria by which they evaluate our ability to get grants, and the NIH is the major grant-giving institution in the United States, and so if it's one of the criteria, they must have defined it. Shaw said, they probably didn't, but they actually did. They did define it. And they defined it as saying, does the project challenge existing paradigms or develop new methodologies or technologies? And that phrase, challenge existing paradigms, leapt off the page of me. Because, to be honest, I think that's what draws people to science. That definition of innovation. You want to be the one that sees the world differently than everybody else and is right about it in a way that can be supported with experiments and lead to new therapies. So that's sort of the working model of the definition for the remaining time in this lecture. And this is the one that I think drives most curious scientific minds. And the origin goes all the way back to Sir Francis Bacon, one of the fathers of the modern scientific method, who said, as the births of living creatures at first are ill-shapen, so are all innovations, which are the births of time. So he dealt with innovation as he was proposing experimental observation, generating hypotheses, testing hypotheses, and creating theories. And then subsequent to him, Louis Pasteur addressed the issue of luck, because if you're lucky, then good things happen to you, and you can create more. But Louis Pasteur said it very famously that luck or chance favors the prepared mind. So that the concept that one innovates as a function of time and preparation and luck come together. And leave it to Albert Einstein, I think, to summarize it in a way that is lasting. And he said, innovation is not the project of logical thought. He actually said, if the original idea is not absurd, then it's not innovative. So the real celebrations in innovation are when the absurd ideas, the illogical ideas, all of a sudden become true because, to continue, the result is tied to logical structure. So that's innovation in science. Crazy-ass idea. Nobody would have thought of it. It doesn't make any sense. At the end of the day, it's proved true because that's really how the world works. So, challenge existing paradigms, look at how the world works, and think about it in a new way. I would like to tell you a story of a patient I took care of with sepsis, as Larry alluded to, but before I get to that, I want to create some historical context and give you an example of a major paradigm shift that occurred, perhaps one of the largest paradigm shifts that occurred in the history of medicine and Western medicine, and then lead you through an example of one that is replacing that replacement in a current version of another paradigm shift. So the ancient Greeks believed that infectious disease, virtually all disease, was caused by humors. And these humors were bodily substances, blood, black bile, yellow bile, and phlegm. And it was very important, they believed, that these materials were present at the proper amounts in the body. Too much of one would cause depression, too much of another would cause insomnia, too much of another would cause heart failure. So what they would do as treatment, understanding that these these humors had to be balanced is they would remove humors, they would do cupping, they would do bleeding, they would do purgatives, or they would add humors, but they would do this by changing diet. So it was really a holistic approach to medicine. Now that approach to treating disease lasted for 2,000 years. And it's the longest standing theory of disease causation in Western medicine. And in fact, it's still being practiced. There are still humoral physician practitioners in the Far East and elsewhere that practice this principle virtually unchanged from Galen. But in the West, this idea was replaced almost overnight by the work of Louis Pasteur and Thaddeus Koch and Bill Rock and others who focused attention on the causative agents of disease coming from outside the body, not inside the body. And their identification of microbes and pathogens turned attention to the fact that things coming into the body from the outside produce disease. And that the immune system and everything we can do medically to prevent this attack and prevent these invaders would be the basis of the treatment in the West. And in fact, this was a major paradigm shift. The idea that bodily humors were not the cause of disease, but rather bacteria, microbes, and pathogens. Shortly before I met the patient I'm going to tell you about, a few people began to think differently about this and make suggestions that I think predicted the future, as I'm going to show you. And one was Lewis Thomas, and this was a brilliant man, a New Yorker, who wrote a series of books, and students of all ages should read this book twice. The Lives of the Cell is one of my favorites of his many that he wrote. And he described what happens to an animal when the animal gets injected with bacterial endotoxin, the toxin that covers gram-negative bacteria. And what he witnessed was the self-disintegration of the whole animal that follows a systemic injection of endotoxin, interpreted as a well-intentioned but lethal error. He talks here about the fact that endotoxin put on a cell in a tissue culture doesn't do anything. The cell is fine. It's not inherently toxic. But endotoxin put in an animal, he says, must look really bad to the animal, because the animal goes into shock, its organs turn black and blue, and the animal dies quickly. It's a self-disintegration. There's something about the molecule that the animal's really not like. And the mechanism of responding to endotoxin is a good one, because you need to know when there's an infection in your body, and your body has to mount an immune response and respond. But this has to be used, he says, with precision and restraint. He's predicting, if you think about it for a minute, he's predicting that it's not the bacteria or the toxins themselves that are doing it. It's the reaction that's killing, in this case, the animal. And he was right. He was like the monk in the cave guarding the manuscript with a candle. He was the only one talking this. He was the only one who believed this. But he would write quietly about this, and it's very interesting to go back and read this now. So that was in the 1970s, early 1980s, at a time at which I was a neurosurgery resident, and Larry only said one thing incorrectly. I'm a recovering neurosurgeon. I swore off it. I don't do it anymore. My name's Kevin Tracey, and I'm a recovering neurosurgeon. And I haven't touched a patient in a long time. But when I was training as a neurosurgeon at the New York Hospital in Cornell, I met a patient named Janet. She was 11 months old, and she had crawled across the kitchen floor while her grandmother was cooking spaghetti. And she crawled under her grandmother's legs, and she tripped the older woman, and the woman spilled the spaghetti boiling spaghetti water on Janice, who became severely injured. She had a 75% total body surface injury, full and partial thickness burns, and was critically ill when I met her in the emergency room at Cornell in May of 1985, when I was at R2 in surgery. She was stabilized in the ER. We admitted her to the burn unit, took her to the operating room on Monday. She did pretty well, until Tuesday night, when all hell broke loose. Her blood pressure plummeted, she went into acute shock, her kidneys failed, her lungs failed, she was on life support and near death. And I had to talk to the family. And the family said, "Doctor, what happened?" I said, "Well, she's in shock." And they said, "From what?" I said, "Toxins." He said, "Well, where did they come from?" I said, "Well, she has her burns, and the bacteria presumably came in through her leaky skin, which is no longer functioning. And the bacteria are there and caused her to go into shock, because endotoxin causes this problem of shock." And they said, "Well, did you find any bacteria?" And I said, "No. Actually, we didn't find any bacteria." "Well, did you find any toxins?" "No, couldn't see the toxins." And for those physicians in the room who had to talk to a family, families who've had to talk to physicians in this situation, the doctors and the families are both acutely aware when the doctor doesn't know what she's talking about. And it's a terrible situation to be in. Because you don't know what you're talking about, the family knows you don't know what you're talking about, and you're just giving the dogma. But I gave the dogma, and the dogma was, it must be this, because we didn't know anything else could possibly do this. And subsequent to this, actually this following summer, I went to the laboratory and began working with my colleagues at Cornell and Rockefeller. We actually discovered that the toxin that caused Janice to go into shock was actually not made by the bacteria, it was actually made by her immune system. Janice made the toxin that put her into shock. And the name of the toxin was TNF, or tumor necrosis factor. And you can see in this slide an experiment that we published in the mid-1980s. We gave TNF to a rat. Now this rat didn't have any infection, or any endotoxemia, or any endotoxin. And within minutes, the rat's blood pressure fell to 0 and it died. And when we cut the animal open very quickly, the heart was still beating, but the lungs had collapsed, the adrenals had necrosed, the gut had necrosed. And it was clear that this product of the immune system, in and of itself, was sufficient to cause a disease in an animal that was indistinguishable from an infection without any infection. And so this was published to much acclaim. I mean, this was in the New York Times, and it was very, very topical because it was unthinkable that the body would do this to itself. Because everyone believed that everything the body did was good for you. So we then went on and said, well, if this is occurring, maybe we can treat it. And we developed a monoclonal antibody, and we did a different experiment. And here we took a baboon and we infused the baboon with bacteria, E. coli. And these were massive amounts of bacteria, and the control animal went into shock and died. And we had built an ICU for this experiment. We had Swan-Ganz catheters and measuring cardiac output, fluid resuscitation. It was a huge deal. And we couldn't keep the animal alive because it went into shock and died. But then we made a monoclonal antibody that blocked TNF, and we treated animals that had bacteria in their blood. But look, they didn't go into shock. And so all these animals out here have bacteria in their bloodstream at 6 and 7 hours. So the bacteria are there, but if you take away the TNF, there's no shock. So this was proof then positive that TNF was both necessary and sufficient to cause a disease that was indistinguishable from acute shock. Now we see this kind of shock every day in the major medical centers in the United States. And the typical patient would be one with fulminating meningococcemia, where there's replicating meningococcus in the bloodstream and in the brain. And these people come in and with hours they go into shock like this and they often die. And this is a TNF problem. Now, it was impossible to translate that, and we wrote about that in these two papers. It was almost impossible to translate that to a treatment for infection because you can see how fast this is all happening. You'd have to basically pre-treat the patients. But it turned out that TNF causing disease wasn't limited to infection. It also causes disease in other chronic treatable diseases, such as rheumatoid arthritis, inflammatory bowel disease, psoriasis, and in fact, this was the first description I'm showing you of a monoclonal anti-TNF antibody therapy. This idea currently is used by millions of patients every year who get anti-TNF monoclonal antibodies. The worldwide market is on the order of $5 billion, and this is a clinically approved drug for the treatment of Crohn's disease and rheumatoid arthritis. Psoriasis is in the offing, and a couple of other indications are under progressive study. So it's fun to talk about hundreds of millions or billions of dollars in annual sales, but it's sometimes more illustrative to look at a single patient because people's lives have significantly benefited from this treatment. So this is a young boy. He's in Stockholm. He's a patient of Wolf Anderson's. And he was going along on the top curve is his height is growing normally, and his weight is growing normally. And he developed rheumatoid arthritis. And he had to be put on steroids. And he developed Cushing's syndrome with weight gain and a moon face, and stopped growing. And then along came this treatment here at Enbrel, which is one of the anti-TNF therapies. And he was able to go off his steroids, he resumed his weight gain normally, he resumed his height normally, and he's now playing soccer at high school at a high level in Stockholm, having a perfectly normal life, taking his Enbrel. So this has been very gratifying to watch happen. Janice, meanwhile, developed another problem. She recovered from the shock, but she developed a new problem. This problem wasn't an emergency. This occurred very slowly over a period of time. She had a little fever, then she had a little kidney failure, then she had another problem breathing, had to go back on a respirator, then her GI tract failed and she got jaundice. And had to be fed with TPN. And now again, the talk with the family. What happened? Well, she has severe sepsis. Well, what's caused this? And we said, well, infection, bacteria. Well, did you find any bacteria this time? Yes, we did. We looked for bacteria, we found some. And we gave her antibiotics, and now they're all gone. Why is she still sick? Well, toxins. And, well, why doesn't she have a shot? Why aren't you running around? Must be different toxins. And in fact, these were the discussions that, and similar discussions occurred today in the ICU, unfortunately, because we still don't understand this completely. But what we did learn is that this problem was in fact caused by a toxin different than TNF, and the toxin was different, so the clinical syndrome is very different. And my colleagues and I at Feinstein approached this problem like thinking, okay, we know patients can come in and have too much TNF or another cytokine, I'm not going to talk about IL-1, which can cause shock and death in hours. But most people who come in with infection, pneumonia or gallbladder disease or perforated appendixes, they don't die right away. Actually, they go along for quite a while, and many of them recover. But some go on to die. And when they die, animals and humans with infection, it's usually later, and it's usually downstream of TNF. So the question we had is, might there be a molecule made downstream of TNF that was actually killing people by causing organ failure? And to look for that, we took immune cells, white blood cells, and we turned them on with either TNF or endotoxin. And we looked at proteins that were released by these macrophages into the extracellular space, and we purified them. And we identified a molecule called HMGB1, which turned out to absolutely fit the bill as a molecule that's produced downstream in infection. It causes organ damage, doesn't cause shock. And when we made antibodies against HMGB1 and treated animals, we could prevent them dying from sepsis. And this has now been done by dozens and dozens of laboratories around the world over the last 10 years, and a number of clinical trials are being planned. But the hope is targeting HMG as a problem of excessive inflammation may go down a similar development path as targeting TNF. That means arthritis, clinical trials are planned, inflammatory bowel disease, clinical trials are possible, and actually even cerebral ischemia. So how do you put this together? Well, this is the paradigm shift. This is the innovation, because we're living through what I call the cytokine theory of disease. Now, the germ theory of disease didn't say that all germs were bad. The germ theory said that in some diseases, in some clinical situations, bacteria or pathogens are both necessary and sufficient to cause diseases, and from understanding that, you can treat them. The same can be said now for cytokines. What happens is patients with injury, infection, or ischemia come to the hospital, and they all look very different. A car crash patient looks very different than a little old lady with pneumonia. But they have in common the fact that their immune system is producing these cytokines, TNF, HMGB1, and others, which in the majority of cases leads to the recovery of the patient. Most people who get sick don't die. Most animals who get sick in nature don't die. In some situations, however, the production of these materials becomes dysregulated, and the response to those cytokines now causes the damage that can kill. And from understanding this simple premise, it's possible to treat cytokines in the clinic to treat human disease. Blocking TNF in arthritis and colitis, blocking IL-1 in arthritis and gout, blocking IL-6 in juvenile pediatric arthritis, and the list goes on and on and on. So this is a valid theory of disease causation that is either replacing or extending our understanding of the germ theory of disease. And we're all living through it. This is happening in real time. In 1992, I moved to what's now the Feinstein Institute, the health system in Manhasset, and had a very simple question. I was starting my lab, and I said, well, this place that the red arrow is pointing to must be really important, because if anyone's immune system is capable of killing the organs and that subject, then the regulation of how the immune system is regulated must be incredibly important. And so I came up with a simple idea to study this that was not innovative, was not paradigm shifting, but it was an idea to make a drug. And the idea was, maybe if we make a small molecule that interferes with the metabolism of the cells that are making the cytokines, we could turn off the cytokine production and put it into clinical trials. And so I won't get into why we picked arginine. That's a story for a longer science lecture, but we decided to interfere with arginine metabolism. Now, arginine, there will not be a chemistry test at the end, don't worry. This is an evening lecture, no tests. This is arginine. And this group is called the guanidinium group, this N double bond group. So we knew that we learned that arginine had to get pumped into the macrophages to make cytokines. So we decided, well, let's make a bulky arginine that gums up the pump. And you can see on the right the bulky arginine. There's 4 of these guanidiniums, 1, 2, 3, 4, but they're separated by this huge space. So any one of them would get in the pump and gum up the works. And now the arginine can't get in the cell. And this was an exciting time. It worked. We blocked cytokines, and we began publishing papers furiously in all of these diseases. We could cure anything in animals that was driven by cytokine production. It didn't matter if there was an infection, peritonitis, sepsis, endotoxemia, meningitis, or Coxsackie, or whether it was a sterile injury with no infection, which is arthritis, colitis, pancreatitis, and the list goes on. And the reason is that by the cytokine-driven diseases, the tissue damage that occurs, whether there's an infection or a sterile injury, both dependent on the overproduction of cytokines. And CNI-1493 was a very good blocker of cytokine production. So we started a company. The company did clinical trials in Crohn's disease, which were favorable in 20 patients or something. And this molecule is now still in clinical development. An oral formulation is being made, and we're waiting to see if it's going to work. But it was a very interesting story. So we thought we had this all figured out. Not particularly innovative, cool, worked, might help people. And then the surprise came. We were working on stroke, cerebral ischemia. And it was fairly logical why cytokines in the brain can kill neurons. And we put the drug into the brain, directly into the brain during stroke. And then, as predicted, very, very small amounts of drug right in the area of the brain turned off cytokines and prevented the damage. But then the surprise came, the chance, the luck, the unexpected. Because we found out that when the drug was in the brain, vanishingly small amounts, that the animal's entire immune system stopped making TNF. And this made no sense whatsoever. There was no logical way that a small amount of material in the brain of an animal would turn off TNF production in the whole body. And this became an area of intense discussion and years of lab meetings, drawing the same pictures on the board. Okay, this is what we know. How can this be true? And what ended up being the case is we ended up cutting things, putting the drug in the brain, cutting the nerves carrying the information out of the brain. We reasoned that the drug in the brain must be doing something to the brain, and the brain was turning off the immune system. And when we cut the vagus nerve, a pair of nerves that runs down the neck, across the chest, and into the spleen, the liver, and all the major body organs, when we cut the vagus nerve, the drug didn't work anymore. So now we realized what was happening. The drug in the brain was somehow turning on the vagus nerve, this major conduit of information to the organs of the body, but never before suspected of having anything to do with the immune system or cytokines. We knew the vagus nerve carried action potentials, electrical activity, that's how nerves work, but we had no idea about how TNF was being turned off by signals in the nerve. So we went back and studied what we had to study about the vagus nerve. And it turns out to be the vagus nerve has a very interesting history. It was involved in a paradigm shift of its own in the early 1900s. Nerves, I don't know how many lay people are actually here, but if there's any, nerves are cells. And they have a cell body, and then typically a long projection called an axon, and a bunch of shorter ones called dendrites. The dendrites receive information from other nerves, and the cell body produces action potentials which sends it down this axon like a wire. And this electrical activity terminates at the nerve endings, and the electrical information is transduced to the responding cell, whether it's a muscle cell, another nerve cell, a heart cell. This is pretty standard stuff. And in the early 1900s, the big fight, the big mystery was, at the end of this nerve ending, when the electricity gets there, does the electricity transmit the information to the responding cell, or does the nerve release a chemical? And the chemical tells the responding cell what to do. There was a huge fight going on for decades. And the answer came from a now famous experiment done by Otto Lowy, who shared the Nobel Prize with Henry Dale for discovering that the basis of neurotransmission was actually chemical, not electrical. And the reason he was able to do this is that on Easter night, Easter Sunday night, 1923, he had a dream. He dreamed the experiment. And he woke up and wrote it down. Now, how many of you keep a pencil and paper on your bedside table? You're all supposed to. I don't. But he did. And he wrote it down. He woke up in the morning, and to his horror, he couldn't read his handwriting. He had no idea what he did. And he knew that this dream was really important. And he couldn't remember the dream, he couldn't read what he wrote. So he spent the entire Monday very upset and went to bed Monday night and had the same dream. This time he got out of bed and went straight to the lab. And what he did was this. He took 2 frogs. The first frog, he exposed the heart, and he electrically stimulated the vagus nerve, and he saw that the heart slowed, because the vagus nerve sends a tonic brake to the heart. When the vagus nerve fires, it slows the heart. Then he removed fluid from that heart that he had electrically stimulated the vagus nerve, transferred it to a heart which he removed the nerve, and that caused the heart to stop. So this proved that the electrical signal led to a chemical or a transmittable factor that he could transfer to the other heart and slow the heart. And this molecule turned out to be acetylcholine, which is shown here, which was isolated a few years later by his co-Nobel laureate, Henry Dale, who isolated it from the spleen of oxes and horses. Oxa and horses. Those are not what he found was that when he purified acetylcholine, AC, when he made it in the lab, chemically made acetylcholine, he could put it in a cat, and you see the blood pressure fall. And it was exactly the same response he obtained from the spleen extract, SE, which was the acetylcholine he purified from the spleen. So this cinched the identity of the vagus nerve slowing the heart, producing acetylcholine through a chemical transmittable signal. This turned out to be, as I'm going to show you in the next couple slides, the fundamental principle by which the nervous system controls all organ function. Organ homeostasis is maintained through reflexes. These were best elucidated by another Nobel laureate, Edgar Douglas Adrian, who wrote a treatise, which I don't necessarily recommend, because it's about 4 inches thick, and it's all about reflexes. So I decided I better summarize it for you here. And what he understood was that a reflex has 3 parts. It has an effector organ, which is typically a gland or a muscle cell. In our case, I'm going to tell you it's a macrophage. It has a conducting path for a conductor leading to that organ, and it has an initiating organ or a receptor whence the reaction starts. So what does that mean? Well, those 3 components mean that your kidneys, your heart, your lungs, and your GI tract, and all your other major organs function without you thinking about them, because they function reflexively. There's a signal that goes up, a sensory nerve. It's processed in the brainstem, below levels of consciousness usually, and a motor response goes back to the target organ to regulate homeostasis around the septum. Now, this was studied since the mid-20th century in virtually every organ system, except the immune system. The immune system's different. The immune system's autonomous. It's not innervated. It floats around. Can't be reflexively controlled. That's the difference. The other organs, pretty easy to study. You can study the heart. You can study the lungs. You can study the gut. You can see them. You can see the nerves. You can stimulate them. And so in the case of heart rate control, heart rate beats along at 72 until something happens, and it speeds up. And when your heart rate speeds up, this change in speed is sensed by the sensory branch of the vagus nerve, takes the information to the brainstem, activates an opposing motor or efferent signal, which slows the heart. The vagus nerve releases acetylcholine, the sinoatrial node, and elsewhere in the heart, slows the heart. We began to think of the possibility that we had stumbled across a reflex to the immune system that was doing the same thing. Because the vagus nerve, it turns out, can sense inflammation, process the information, and send a signal back down the vagus nerve to release acetylcholine to turn off cytokine release, which is exactly what you would want to do if you had a little inflammation. If you have a little inflammation in your body, you want it controlled. You want the brain and everything else to be slowing it down to the right amount, so it doesn't spin out of control like it did in Janus. And in fact, that turns out to be exactly the case. We published many papers on this now. This one was the cover of JPM a few years ago by my colleague Jared Houston, who's now a professor of surgery out at Stony Brook. He was in the lab for 3 or 4 years. And what Jared did is he electrically stimulated the vagus nerve here in the neck, and then followed the signal to the spleen. It turns out that the spleen is the organ that makes most of the TNF that we've been studying. And that was very convenient for us because there's only one nerve to the spleen, one artery, and one vein. So now we could follow the whole system and look at what was happening minute to minute in the spleen. And we could stimulate the neck and cut and see no signal. So we could lesion distal to that, lesion distal to that. We could actually even put lesions in the spleen. And when you do this, what you see is very dramatic. So these are spleens. This is a former grad student, now graduating postdoc, Mauricio Rosso-Spina. Mauricio took spleens and he stained them for cells that make TNF. So each red dot in the top 2 panels are individual cells that are making TNF. And we could follow the TNF out the splenic vein. And this is the TNF that shows up in the bloodstream. Showed up in Janice's bloodstream during shock. It shows up in the bloodstream of patients during cyclic shock. And shows up in the joints of patients with rheumatoid arthritis. When we turned on the vagus nerve stimulator, those cells stopped making TNF. 90%, 95% of them turned off. This really demonstrated clearly the ability that the immune system can control minute to minute what the immune system, the nervous system can control minute to minute through action potentials what the immune system is doing. This has been a big surprise. And it's become extremely interesting recently. We're in the process of publishing this paper, which I just show you because it's a beautiful picture. This is a lymphocyte in the spleen. This is the nerve. Now we know the action potentials that travel down this nerve originate in the neck, the brainstem. This nerve is connected to the vagus nerve. And where the cell turns yellow, that's actually a blend of red and green because that means the nerve ending is touching the cell in the spleen. So, who cares? It's interesting science. Well, actually, this works like CNI 1493 did. This works in all the same diseases. We are busy now implanting electrodes on vagus nerves of animals, and perhaps next week, people, to turn off inflammation. This is an abbreviated list. I could have shown you the same list. I could have shown you the same list as I did before for CNI 1493. But inflammatory bowel disease, arthritis, myocardial infarction, pancreatitis, psoriasis, all of these diseases are treatable in animals by simply turning on the vagus nerve. We put a pacemaker-like device in, fire the nerve sends a signal to the cells making the cytokines in the spleen and elsewhere, and shuts off the inflammation. So, in the interest of full disclosure, I started another company to manufacture devices and test them in people for these diseases, and that company is called Septone. I'm a consultant to the company now, and that company has now gotten approval in Holland and Bosnia as of last week to begin clinical trials on rheumatoid arthritis patients. And the idea is we may be able to put a device in people, eventually under local anesthesia, if not percutaneously, that will stimulate the vagus nerve and allow these people to stop taking anti-TNF, perhaps. Anti-TNF currently costs between $20,000 and $30,000 a year. There's a black box warning on it because of immunosuppressive side effects. And we wonder if this device, which is known to be non-toxic and safe for a number of reasons we can talk about if there's any interest, will actually replace the toxic drugs. Time will tell. We have no reason to suspect any untoward side effects. There's a large clinical experience with stimulating the vagus nerve for other reasons. And our animal models indicate this is very safe. The question really remaining is, will it work? We'll know soon. We're going to start, as I said, perhaps as early as patients are being screened now for surgical implants. So that, I hope, will be a major challenge to existing paradigms. It works in animals beautifully. I want to show you this. These are rats with arthritis. The arthritis is actually caused by immunizing the rats with collagen, which causes this immune reaction against the collagen, which then moves to the joints, and these animals develop polyarthritis that's virtually similar in many, many ways to rheumatoid arthritis in patients. And this arthritis score on the top in in the animals, untreated animals, these are very seriously impaired animals. They can hardly walk to get their food. When we implanted vagus nerve stimulators and turned it on in blue, both groups have vagus nerve stimulators, but we only turned it on in the blue guys. We could treat the animals once a day for 30 seconds to a minute and block the disease from day 11 to day 16 in an effective amount that was quite comparable to how anti-TNF works in the same animal model. So time will tell if this will work. So what's next to conclude? So I love this. There's two cavemen. I think one's at the top of the mountain, and the other walked up to ask him advice. Something's just not right. Our air is clean. Our water is pure. We all get plenty of exercise. Everything we eat is organic and free range, and yet nobody lives past 30. And it's very interesting to look at the epidemiology. The death rate from pneumonia since the introduction of antibiotics has changed very, very, very little. A few percentage points. A little known fact about antibiotic resistance and all these problems. So, one way to challenge the existing paradigms is to think about what the next cool thing to do would be. And scientists are often loath to do that. It doesn't pay well. I mean, to really predict the future, no one's going to give you grants to do that. The NIH will only give you money for what you've already done. Industry won't do it. Ideas, I like to say in lab meetings, are cheap. I really don't care about ideas. I want to hear an idea that leads to an experiment that you can do tomorrow that will address the idea. That's invaluable. That's innovative. That's how you challenge an existing paradigm. Cancer's been cured 1 million times in 1 million different people's minds. But we don't have a cure yet because the cure's there, but how to make it is unknown. And so ideas are cheap. I don't care what you're taught elsewhere. Ideas are next to worthless. But an idea that you do the experiment and it works is infinitely valuable. Infinitely valuable. In a way that you can't put a price on financially. So with that long disclaimer, I am going to tell you what I think I want to do next. Which will probably be all wrong, but today sitting here, some of it's pretty interesting. So I went to the mountain and met the Dalai Lama. This is me here on the left, and this is him there on the right. This is his translator in between. Now, I didn't climb a mountain. I drove up to his compound near Woodstock, Benicia, New York, and spent 2 days talking with other scientists. It turns out the Dalai Lama is convinced that his belief system is correct. His belief system is very similar to the ancient Greeks. And he believes that their approach to medicine is probably better for preventing diseases. But as he said at the meeting, that if he gets in a car accident or needs a surgical problem, he goes and finds the best surgeon he can find, trained in the West, and does whatever she says. That's appropriate. But we got to talking. I presented a summary, and my colleague Liz Blackburn, who was also there, presented the other half of the summary. And then he asked me a question. And he said, this vagus nerve, where is it? I said, well, there's 2 of them. They go down the neck, across the chest, into the spleen and other organs of the immune system. He goes, is there 1 or 2? I said, there's 2. He said, is it in the front or the back? I said, it's in the front. He nodded. And that was that. So afterwards, a monk comes up to me in a long orange robe, striking. And he says to me, do you know why His Holiness was asking you that question? I said, no, I have no clue. He said, well, it turns out there's an ancient Indo-Tibetan meditative practice in which the practitioner envisions a field of blue energy over their head, which they then channel into 2 beams down the neck, across the chest, and into the abdomen. I said, who? It's like, you can't make this up? They've been doing it for, 3,000 years. And it was very interesting. There was a bunch of guys from ivory tower academic places who are interested in acupuncture, meditation, and cognitive relaxation therapy, and you read their papers in all the journals, if you read those journals. And they were very upset to hear my talk, because they came up to me and said, they were arguing with me why this would have no relevance, despite the fact that all of these things on this list, including fish oil, increase your vagus nerve activity. So the more fish oil you take, the more vagus nerve activity you have, the slower your heart rate is, and the more protected you are against inflammation. Same is true for meditative therapies, cognitive behavior. All of these things in humans turn on your vagus nerve, which is exactly what you want to do. You want to be healthy. Turn on your vagus nerve, turn off your inflammation, slow your heart. We're actually doing a study with my colleague at Columbia Presbyterian, Richard Sloan. We're actually doing a large prospective, multi-million-dollar trial on exercise. We're taking couch potatoes, and we're making them fit, and we're following their vagus nerve activity and measuring their inflammation. Because this may explain why exercise is protective against heart disease, which nobody knows why it is. So that was very interesting. That was my trip to the mountain. But the idea that this could explain this was offensive to the sort of academic professor types. It was embraced by the Dalai Lama and by his people. The Dalai Lama says, I don't care how these things work. If they work and Western science disproves something we believe, I'm going to change our belief system. So the point is, this is an area that can be studied in the future. I'm not going into this much. I just put it up there because I hope other people go into it. What I want to go into is this. This is an eye for the lay audience. See, eyeball. You don't see with your eye. You actually see with your brain. The eye simply gathers information from the environment. It gathers, in this case, light, which activates basically nerves in the back of the eye, and those nerves send electrical information to the brain, and the brain sees. This has been known in animals for decades. Nobel Prizes have been given over it. But it was the best example is actually very modern. There's now a machine that works in people, which is basically a TV camera connected to which collects light from the outside world, turns it into electrical information. And there's now a TV camera that takes that electrical information, puts it on a little grid, a little membrane, which patients can wear on their tongue. People with damage to their eyes, but intact occipital lobes, which is what you see with, can see. And they learn very quickly. This device has been through a series of successful clinical trials. It's gotten regulatory approval in a couple of countries. It's I believe it has preliminary regulatory approval in the United States. So if the brain works, it doesn't matter how the information about the outside world gets to it. Once it's in the brain takes care of relaying it to where it needs to go to be processed to see. You see with your brain. The eye is a signal transducer. So if that's true, and it is, then why wouldn't this be true? Why wouldn't the immune system be a way of seeing the environment all day long? We know that cytokines, bacteria, and products of pathogens turn on neurons. We know that those neurons track through the vagus nerve and other nerves to the brain and change the behavior in the brain. We've measured it, we visualized it, we can see it. Not just me, but laboratories around the world. These are all facts. So if it's true that you see with your brain, because the brain is processing all the information from all these individual beams of light, why wouldn't it also be processing all the information from the immune system? It must be. It must be. So I have now money from the Department of Defense, of all places, to actually study this big project with them, through DARPA. And the idea is that the brain, if this is all true, the brain must know what the status of the body is. And so what we're proposing to do is decode this and develop a grid that will sit on the brain so that when the injured warfighter comes in, their helmet has a receiver in it, or an EEG type of ring, that is plugged into a computer, like when you drive your car into the gas station. The mechanic doesn't even look at you. He plugs it in, the computer into your car, and he looks at the computer just like this one. And the computer says, no pressure in the sixth cylinder, low tire in the low air pressure in the left rear tire, and you need an oil change. It's all right there on the computer. The computer in the car knows what's wrong. So the theory that we're studying now with this project is that the brain would know if we knew how to deconvolute the signal. The brain knows that there's a hole in the liver. Sorry. Liver. Be new anatomy, Dave. The brain knows there's a hole in the liver. The brain knows that there's an abscess in the lung. The brain knows what the body temperature is. All that we think can be pulled out. And so to do the experiment, we're literally this week have been injecting inflammatory agents into the nerve endings and recording the signals going up, because that's the first step in deconvoluting. If you think about it, this is not particularly innovative. actually, right? We're going to stimulate a nerve and record. And in doing that, I showed you for 150 years. We're going to stimulate and record. But what would be very good if we can deconvolute the signal and and say this electrical pattern in the brain means there's a hole in the loop. So that's what we're doing for the future. I don't know if it's Janice was getting ready to be discharged. She was going to be sent home from the hospital. And I was walking by the door of her room at lunchtime, and she was drinking a baby bottle in a rocking chair as the nurse was rocking her in a rocking chair, and she rolled her eyes back and died. I was the first person there, 3 steps away, gave her mouth to mouth, called the code. The code was flawless. My senior resident, Al Pfister, was there, coincidentally, a few steps behind me, also unplanned. He was a cardiac surgeon in training, floated a cardiac pacemaker. They had just been made available to the burn unit there. I'd never seen one before. He floated it and got it in on the first pass. It was a flawless code, and we never got captured the heartbeat. She was dead. And when I got the ME report back from Bellevue a few weeks later, I was aghast because there was no cause of death. She didn't have widespread infection. She didn't have widespread inflammation. She didn't have a PE. She didn't have heart disease. And I knew that they had written down on the death note, they had written down on the autopsy, the words they'd taken off my progress report, my death note, cardiopulmonary arrest. And so to this day, I don't know what she died of, but she haunted me. She's the only patient I ever gave mouth-to-mouth to. She's the only patient I ever had nightmares over. And to this day, I don't know. I think that she may have had a massive impairment of her ability to regulate cytokine production. And there are other cases of this where different types of neurogenic failure can lead to sudden death. I don't know. But I think her legacy goes on, and I think that her the questions that we couldn't answer to her family then, we can answer very differently now. I think that there's a lot of evidence for keeping your brain smart. There's a lot of evidence for having a positive outlook. All these things increase the activity of your vagus nerve, which slow inflammation. Same for fitness, which may influence your ability to sense the activation of the immune system. And finally, together, this knowledge, I think, will drive the continued development of drugs for quote-unquote healthy immunity based on the cytokine theory of disease. We have clinically approved drugs that target individual cytokines. We're developing drugs, I didn't go into it today, at this nerve ending. We know the receptor, we know the acetylcholine target, we've isolated the gene product, and we make highly selective pharmacological agonists that do the same thing as vagus nerve stimulation. And the vagus nerve stimulator itself is in development, and I hope it will work. And with that, I'll say thank you. Dave said we can have questions. Ronnie?
Yeah, I have a question. Knowing what we do about the sort of variability from person to person in terms of immune responsiveness and things like that, or immunity and developing that, is there any evidence for differences in sensitivity or variability in the connection between the vagus, the macrophage, and the release of TNF?
Can you repeat the question?
Yes. So basically the question is, what do we know about variability between the end of the vagus nerve, in the spleen in this case, and all the cells passing through it? It's a billion-dollar question for a number of reasons. First of all, it's a brand new field of study. There's almost nothing known about it. But if you look at what has been done, it's very interesting. It looks as if the whole thing is plastic. So the nerves in the spleen are constantly changing during inflammation. They're dying back, they're growing new, and at the same time the cells are turning over. The spleen gets 20% of cardiac output every minute. And so this is actually an opportunity for specific neural signals ending in the spleen to educate the whole circulating population of the immune system, not just the macrophages and the T cells that are fixed in the spleen. So it's an area of active investigation. Betty Diamond and I were actually meeting before I came over here today, and we're looking at that in the context of immunization and B and T cell responses. We kid each other. Betty tells me that the B cells, she's an antibody person, the B cells are telling the nerves what to do, and I tell her the nerves are telling the B cells what to do. But nobody knows which is true. If you look in arthritis models, which is really interesting, and this has been done by Rainer Straub's group in Heidelberg, he basically gave the same arthritis model I showed you, and then looked at the nerves in the spleen, and what he saw was, right before the animals get really sick on that up curve, and they get severe inflammation, all the nerves in the spleen die back. So right when you want those nerves there to turn everything off, they're gone. Where did they go? Why did they go away? What turned them off? It's a really important question. We don't even know if it's a classic synapse. It may be that the nerve endings in the spleen release neurotransmitters which perfuse all the cells in the spleen. We're studying that possibility too.
So along those lines, what is your idea about what the afferent lobe that reflexes? If it's a spleen part of it is crossing the blood-brain barrier?
So the afferent limb of the reflex has been studied a little bit, and there's let me back up a second. So the circuit is sensory, processing, afferent, processing, and efferent. The efferent side, we really have mapped. We know the microbiology, we know where it is, we know the neurophysiology I showed you at a high level. The sensory side is a black box, and the processing brainstem processor is a black box. There is some very interesting data on the sensory side, done primarily by Linda Watkins and Steve Meyer. What they did is they gave IL-1 to the abdomen of a rat, which caused a fever. Then they cut the vagus nerve, and there's no fever. So the vagus nerve is sensing the IL-1. What you're asking is another billion-dollar question. How does the vagus nerve see IL-1? One possibility is that there are receptors for IL-1 on the nerve endings, and there's some evidence for that. The other possibility is that IL-1 is interacting with cells in the region of nerve endings which make neurotransmitters which are sensed by the sensory nerve. So it's also very related to the project I told you about DARPA, because if you really understood, you could imagine that the reflex could be impaired because there's no efferent signal, or it could be impaired because it doesn't sense anything.
What if it's in the solitary current?
So the action potentials have been mapped to the nucleus tractus solitarius and to the DMP. But what the whole circuit is, which came first, chicken and eggs, not known. We've recently done some micro-PET studies and shown that if you actually use a pharmacologic, I never told you in the story, CNI 1493 works because it interacts with the muscarine receptor type 1, which turns on the descending cholinergic signals that turn on the vagus nerve in the periphery. And that pathway you can visualize as micro-PET, and that actually culminates, you can actually see on micro-PET the vagus nerve on the left side turning on. So we're moving into the nuclei next.
So in recent years, people have been discussing depression, clinical depression, as an immune response, more and more. And I've read at least one or two papers that have described experiments of using vagus stimulation to treat depression fairly well. So what I think of is how, and we usually have depression as like a right frontal type illness, usually. So have you encountered any evidence that there's communication between these sorts of deeper brain reflexes and more frontal processing?
So yes and no. It's a very controversial point about the mechanism of action of vagus nerve stimulation in depression. And as you know, it's a problem. It's a complicated story. It was quote unquote approved in the United States, but the insurance companies don't pay for it because the data are so soft that it works. And it does work in some patients, but there's no way to predict which patients it's going to work in. The response rate is what, 20-30%. So it's not great. But the ones it works in, it works great. The story to me is extremely interesting. Not from the dogma is that the signal goes up into the brain. Okay, but the data are that if you really look at the data, again, this goes back 30, 40 years. If you really look at the data for inflammation in patients with depression, what they actually have is they have an overproduction of cytokines in response to minor stimuli. Now, cytokines cause behavior. We tend to think that the brain is being in charge. It's not. The immune system is in charge. If I inject you with cytokines, you will change your behavior. You will develop a fever. You will develop anorexia. Patients with cancer who need to get cytokine infusions for hepatomas, use interferon for hepatomas and some melanomas. The oncologist does two things at that appointment. They say, if you're going to go for this, give you a prescription to go see the infusionist, and they give you another prescription to go see the psychiatrist first, because they now pre-treat these people for depression because the cytokine infusions cause depression. So the alternative mechanism by which the vagus nerve stimulator may work in some patients is that it's sending a signal to a spleen and it's resetting or suppressing the cytokine production, which is causing the depression in the brain. So let me answer it another way, which is not a criticism, criticism of anybody or anything or any company, but the methods to use vagus nerve stimulation for depression and epilepsy date back to 1895, when it was observed accidentally or paradoxically that rubbing on the carotid of patients with seizure disorders would stop some seizures. And that's the science behind how vagus nerve stimulation works in epilepsy. Cyberonics has now sold 60,000 of these devices, and it does work in about 30% of patients with epilepsy. But these pathways I drew for you, no knowledge how that works in depression or epilepsy. And people are getting more interested in it. I think in both cases, cytokines could play a role. There's clear evidence that cytokine overproduction causes depression, and there's clear evidence that cytokine overproduction desensitizes neurons firing, which contribute potentially an epileptic form situation. Ready?
So if you can measure the vagus nerve reflex and quantify it, can you then screen people and say whose system is working well and who's at risk of having a problem?
We patented that question 6 or 7 years ago. It's incredibly important. What Betty's saying is, if you know that someone's vagus nerve is working normally, despite the fact that they have an inflammatory disease, you wouldn't want to stimulate their vagus nerve. They could have, this isn't the, let me be very, very clear about something. This is not holy water. I'm not selling you anything. This is not the only way that the nervous system controls the immune system. This is an example of a prototypical reflex. I think 10 years from now, there'll be 10 more circuits that are mapped with individual nerves controlling discrete immune functions through discrete neurotransmitters through discrete receptors. This is just as one that we could map and we understand. But if you understand it, what Betty's point is that you wouldn't necessarily want to implant a nerve stimulator in someone who didn't need it. And so the way that what we propose to do, what we have proposed to do in a patent and are developing the methods to do, is you want to understand what the resting vagus nerve activity is in a volunteer. And if they don't need the device, they don't get enrolled in the trial. So you do two things. You prevent any unnecessary surgery, but you also improve the ability of your trial work. Now we don't have that yet, so we're also developing a preclinical tester in which we will take 20 people with inflammation and we'll pre-treat all of them with a non-surgical tester through the base of the skull, and only then implant the ones that respond. It's the other way to do what you said. But hopefully that will enable us to erase the clinical trial results. There's beer outside, Jesse. You really want to ask this?
Well, let's get over. I was just mentioning, you're so big on sepsis survivors. Just a word about those I thought was a...
Oh, Jesse, sepsis survivors. Yeah, I mean, so that's another futuristic project. 200,000 people a year die from sepsis in the United States. It's the leading killer of hospitalized patients in the United States. It's the leading killer of hospitalized patients in hospitals of the world. And we have not been able to treat it before Janus or after. And there's a number of reasons that make it exceedingly difficult to think we're going to be able to intervene in a timely enough way to significantly change the mortality. But people are still trying, including us. On the other hand, we became very interested recently in the statistics that 800,000 people a year survive severe sepsis. 4 times more live than die. And half of those people die in 5 years or less. Those are huge numbers. Huge numbers. That means 1 million people are dead a year because they survived sepsis in the past 5 years. So we've been, and they're significantly cognitively impaired, and we found now, and so we set up animals, for many, many years we've been studying animals with sepsis. Now we stopped studying the ones while they were sick, and now we're studying the ones that live. And what we see in these animals that survive sepsis is just like their human counterparts, they have major cognitive impairments, they have difficulty learning, they have completely abnormal stress responses, and it appears, like I alluded to in the end of the story about Janus, that their vagus nerve function is totally deranged. It doesn't work. And so this is the future. I really can't comment on where it's going, but I think we're literally shifting the interest in my lab away from trying to treat the acute problem in the hospital towards dealing with understanding whether we can actually treat the people in the hospital who look like they're surviving in order to give them a better quality of life after they go home. Yes?
Any indication that there are downsides to vagus nerve stimulation?
Another $1 million question. I mean, The scientific and medical answers we don't know until we do the clinical trial for this disease. In 60,000 patients implanted so far around the world for epilepsy treatment, the side effects are pretty much limited to mechanical problems where the nerve gets pulled on or the device gets infected. The infection rates are not higher with this device so far than any other implanted device in knee or hip or anything else or stent. And so we don't have any evidence to think that the device will have an adverse risk profile. It's never been put in people with inflammatory diseases like ulcerative colitis or rheumatoid arthritis. And they may have other risks added to, they may come out when we do the trial. So in a clinical trial, you know, with the rule in pharmacology and in drug development, device development, everything's toxic. At some point, everything we do in medicine is toxic. Whether this will be toxic or not, I don't know. I hope not. In animals, we're not seeing any significant immune impairment. And the very important point about your comment is that as opposed to anti-TNF, Remicade, Enbrel, and these other antibody-based drugs that neutralize 100% of the TNF that's available. An antibody doesn't discriminate. If there's TNF there and an antibody there, it's wiped out. The antibody immunosuppresses the TNF. These animal models, I didn't have time to print all the data, obviously. The device only blocks about 60% of the TNF that's produced, which is just enough to bring it from the toxic level down to the non-toxic level. It doesn't immunosuppress. So we're hoping that we're not going to see any problems in the clinic. I don't know. We'll know in a year, or maybe we may know sooner. Rita.
When you injected CNI 1495 into the brain, was there a specific area of the brain that you injected it into, and why?
We injected, so the question is, when we injected CNI 1493 into the brain during the stroke studies, did we put it in a specific place? Yes, we put it into the side of the brain that we'd given an infarct. We had cut the cerebral artery on one side to give a stroke on one side, and that's the side we put the drug on because we knew there was TNF being overproduced there. We wanted the drug there in very small amounts, just sort of bathing the region. And it was a total, there was no anatomical basis for us to think about why the signal would come out of there from that. It was a big surprise. Maybe I'll just close with this, with the real story behind CNI 1493. CNI 1493 was the 14th molecule that we screened as an arginine uptake inhibitor in 1992 by Marina Bianchi and Carlos Ramos. And so it should have been named 1492. But we didn't, but Columbus, all the baggage with competing with Columbus, so we took artistic license and we named it 1493. And like they used to say in Saturday Night Live, 1493 been very, very good. So, thank you.